My name is Dr. Ciara Freeman. I work at the Moffitt Cancer Center in Tampa, Florida, and at this year's annual American Society of Hematology meeting, on behalf of all of my co-investigators and the study team, I presented the preliminary results of a phase 2 registrational study of Anitocabtagene Autoleucel for the treatment of patients with relapsed or refractory myeloma, the very preliminary results of the IMMagine-1 study. So Anitocabtagene Autoleucel, or Anito-cel, is a BCMA targeting CAR T-cell therapy with a 4-1BB and CD3 zeta co-stimulatory domain, but is unique from other constructs in its binding domain, which is a small D-domain, entirely synthetic protein that has a triple alpha helical structure and a stable internal hydrophobic motif. And because of this, it facilitates this high transduction efficiency and more CAR expression on the surface of the cell, resulting in a lower total cell dose. Because of that, the CARs are very stable and potentially lack tonic signaling because of this rapid folding, lack of disulfide bonds, and this hydrophobic core that I mentioned, in the way that the D domain folds itself, and therefore has a number of additional features that may be advantageous. One of them being it has this faster off rate and high CAR surface expression, and this may allow for optimal tumor killing without prolonged inflammation and exhaustion of the cells. By way of background, this data was also presented at the meeting by my colleague, Dr. Bishop, showing the longer-term follow-up of the initial phase 1 first-in-human study in a very high-risk, heavily pretreated population, just showing that with a median follow-up of over 3 years, patients who had had 4 or more prior lines of therapy were able to achieve a complete or stringent response, complete response in 79% of those treated, with a median progression-free survival of 30 months. So that means at 30 months, 50% of patients still had not progressed or died. And that was out at 34 months for those patients who achieved a complete response. Overall survival had not been reached at that time of follow-up, with similar efficacy seen across high-risk subgroups, in particular in those who had extramedullary disease, and this is quite enriched in this study. And importantly, they had not observed any late onset or non-eye cancer neurotoxicities, including Parkinsonism, cranial nerve palsies, or Guillain-Barré. So this was the trial that was presented, the phase 2 single-arm study of Anitocabtagene Autoleucel in patients who'd had at least 3 prior lines of therapy, including the major classes of drugs, so immunomodulators, proteasome inhibitors, and anti-CD38 antibodies like daratumumab or cetuximab. They had to be refractory to their most recent line, have measurable disease, a good performance status, and good organ function, as is often required for all trial participants. The overall response rate was the primary endpoint as evaluated by the IMWG criteria, but the data that I presented and will share with you here was investigator-assessed responses. So that's important to note, with the data cutoff of the end of October, which was very preliminary at the time of this presentation. At the time of the cutoff, 129 patients had been enrolled and leukapheresed and comprised the total population of the study. 11 discontinued prior to lymphodepletion, so that's the chemotherapy that's given before the CAR T administration, for reasons that are outlined here, the majority because of either withdrawal of consent or disease progression, as can commonly occur in this patient population. This resulted in a total dosed population of 117. Based on the data cutoff of the end of October of 2024, 98 were evaluable for safety, who had at least 1 month of follow-up, and for efficacy, at least 2 months of follow-up were required, and for that we had a population of 86 patients. These are the baseline characteristics that were very similar between the safety and efficacy evaluable patient population. Important to note that a significant proportion of patients were over the age of 70, as commonly is the case in patients with myeloma with relapsed and refractory disease. They're often older, and a third of the patients were over the age of 70. In addition, we treated 9% of patients who were African American. This increased towards the end of the trials. We enrolled more patients who were African American towards the end. And of course, patients had good performance status, A significant number had extramedullary disease or high-risk cytogenetic features, and of course, all were refractory to their most recent treatment line. We were able to dose up to 10% as an outpatient by the end of the trial. 8 and 6% are shown here. So at a preliminary time cutoff with a median follow-up of 9.5 months, the overall response rate was 97%, and the complete response rate or CR rate was 62% in this patient population. What is interesting that of those patients who had known evaluable for MRD assessments, that's minimal residual disease assessment, 93.1% of them were MRD negative at a sensitivity of 10 to the minus 5 threshold. And the median time to achieving MRD negativity was just 1 month. Again, this is a very preliminary data cut, but at this time point, the progression-free survival estimates at 6 months were 93.3%, and at 12 months were 78.5%. And again, these will probably mature with more follow-up, with an identical overall survival estimate at both time points. Cytokine release syndrome was the commonest toxicity observed in 68% of patients who had grade 1, 13 at grade 2, and a single grade 5 event. Management was as per protocol and in line with the usual criteria. No patients received prophylaxis for cytokine release syndrome. That's important to note. And 17% of patients had no CRS event at all. The majority of events resolved within the first 10 days, and most patients were treated with tocilizumab and or dexamethasone, with the additional measures shown here. There was one grade 5 CRS patient that unfortunately succumbed to CRS. This is a patient, 76-year-old, who had rapidly progressive disease occurring between screening and baseline, and unfortunately did not receive bridging therapy, just highlighting the importance of taking patients forward for CAR T-cell therapy with well-controlled disease. As you can see here, ICANS was not common, occurring in just 9% or 9 patients treated, all cases resolved. And as of the time that these data were reported, there had been no non-ICANS delayed neurotoxicities reported across the entire trial portfolio. Treatment emergent adverse events are shown here. The common grade 3 events were cytopenias, as we would commonly see in patients treated with CAR T, with the other events shown here, again, that would be very much in line with our expected treatment in patients like this who have relapsed refractory disease treated with CAR T. There were unfortunately 3 deaths that occurred, one from CRS, as I've already mentioned, one event of a retroperitoneal hemorrhage that occurred in a patient who had evidence of hemophagocytic syndrome, or IECHS, and had a bone marrow biopsy performed in the setting of coagulopathy to confirm the presence of hemophagocytosis, and unfortunately had a massive hemorrhage occur due to that biopsy. There was also another patient who had an invasive fungal infection, again, just highlighting the importance of careful monitoring, prophylaxis, and aggressive surveillance of these patients for infection. So in conclusion, Anito-cel, with its novel synthetic compact D-domain binder, facilitates high transduction efficiency and CAR positivity with high density of CARs on the surface of the cells, demonstrated deep and durable efficacy at this early and preliminary time point with an overall response rate of 97% and a complete response rate of 62%. MRD evaluable patients showed high levels of MRD negativity at a threshold of 10 to the minus 5. And of course, more prolonged follow-up is needed for this patient population. But at this time point, 12-month PFS was 78.5% and overall survival was 96.5%. The safety profile is certainly as we would expect in this patient population, but importantly, there have been no delayed non-ICANS neurotoxicities reported to date across a patient program that now spans over 150 patients. Importantly, the IMMagine-3 trial, which is a large global phase 3 study, randomizing patients who have had 1 to 3 prior lines of therapy, which includes an anti-CD38 monoclonal antibody and an immunomodulator, is now open to accrual and actively accruing patients to be randomized to either standard of care or Anito-cel. And this trial is open here at Moffitt and in many other centres around the globe, and we're very excited to see how this performs in a randomized setting. Of course, none of this is possible without the bravery of patients who commit themselves to enrolling in clinical trials and scientific advancement, and of course, the entire team of co-investigators across all of these amazing sites and the study teams who go so far to make sure that the data is appropriate and accurate. And so I'd like to thank everyone that's participated in the study.